Assalamualaikum and a very good morning. A warm welcome to all participants from Malaysia, participants from SEAMEO Network and affiliated countries to IMR-SEAMEO webinar series 2022. The theme today is whole genome sequencing analysis for comparative investigation of bacterial genome diversity. Our presenter today are from Institute for Medical Research in Malaysia. I am Dr. Roszilawati Hashim, the head of Bacteriology Unit, Infectious Disease Research Center IMR, will be the moderator for today's session. Ladies and gentlemen, this webinar series was initiated by the aim in mind to share the knowledge, discuss topics on biomedical research, and to present research outcome. Before we begin our presentation, allow me to announce a few housekeeping messages from the Secretariat. The first is we will not be taking questions during the presentation, but participants may post their questions using the link that has been shared in the invitation email and YouTube channel. Number two is the question and answer session will be conducted at the end of the third presentations. And number three, the attendance link is also provided for those who would like to receive an e-certificate of attendance. You may find the link in the chat box of the YouTube channels. Ladies and gentlemen, for today's webinar, we have three speakers. Our first speaker is Dr. Siti Roszilawati, who is a clinical microbiologist in Bacteriology Unit, Infectious Disease Research Center, IMR. She holds a medical degree from International Islamic University of Malaysia. In 2011, she was awarded Master in Pathology from University Kebangsaan Malaysia. And in 2019, she obtained a PhD degree in Molecular Microbiology from Technical University in Braunschweig, Germany. Her research area of interest are leptospirosis, tuberculosis, and zoonotic diseases. She has published papers in international peer-reviewed journals. For today, Dr. Siti will be delivering a presentation entitled Complete Genome Sequencing Analysis of Leptospira interrogans of Different Virulence. Dr. Siti, the screen is yours.
Assalamualaikum and very good morning. Thank you, Madam Chairperson and the Organising Committee of IMR-SEAMEO webinar for giving me this opportunity to present the findings of my study entitled Complete Genome Sequence Analysis of Leptospira interrogans with Differential Virulence. Leptospirosis is the most widely spread zoonotic disease worldwide. The highest disease burden are in the temperate and tropical region, which the case fatality rate range from 5 to 30%. The causative agent is Leptospira species, which can be found in the environment and also the kidney of carrier animals. Human can be infected by direct contact to the carrier animals as well as direct contact to the water and soil contaminated by their urine. Leptospira species are vastly diverse bacteria with more than 21 species and three main phenotypic features, namely pathogenic, intermediate, and saprophytic. They can also be classified based on heterogeneous antigenicity of the lipopolysaccharides, which give rise to the serovars and serogroups. However, the serogroup can have some typical carrier animals, however, this is not definitive. The clinical features of leptospirosis may range from influenza-like illness to multi-organ failure, especially in lung, renal, and liver. The severe form of illness are Wills disease and severe pulmonary hemorrhage syndrome, which the case fatality rate may be higher than 50%. Until now, the genomic makeup and antigenic variability are poorly understood for Leptospira, as 40% of the gene encode hypothetical protein that is unknown in function, and there is high level of functional redundancy in the genes of pathogenesis, and the gene encoding the ability to adapt in the host and environments are still dubious. Thus, this study is being carried out to address the gaps. The general objective is to determine complete genome sequence profiling of 6 clinical isolates of Leptospira interrogans in Malaysia. The specific objectives include to determine the most and least virulent strain by clinical evaluations and animal study. Second is to determine the phylogenetic relatedness using molecular typing, i.e. the WGS typing. And thirdly is to carry out comparative genomic characterization and analysis. For the first objective, around 2,000 blood samples of acute leptospirosis suspected patients were collected and cultured for leptospira, and a positive isolate will be traced for the medical record, and the isolate will then be subjected to virulent tests in animal model, and the findings of histopathology will be the confirmation. And for the second objective, serotyping were carried out to determine the serovar and serogroup, and next whole genome sequence were performed, and the isolate were molecularly typed for MLST, CG-MLST, and SNP-based genotyping. And lastly, comparative genome analysis were carried out using bioinformatic tools. For the first objective, around 6 clinical isolate were collected within 2 years. That is comprised of 5 isolates from patients in Hospital Tengku Ampuan Rahimah, and also 1 isolate from a Dutch patient who fell ill after travelling to Langkawi. And this table of clinical features summarizes the clinical features of the 6 patients, where the age, all patients were male, and the age range is around 15 to 50 years old. And all patients had history of exposure except for 1 patient, that is 1548. And period of exposure ranged from less than 1 week to 3 weeks. And 4 patients were admitted to ICU as they presented with signs and symptoms of septic shock. 3 patients had jaundice and acute kidney injury, and 1 patient had pulmonary hemorrhage. So a severity score were calculated based on the clinical features and laboratory findings, and the result showed that patient infected with Sri Lankawi had the most severe acute leptospirosis manifestation, and this is followed by patient in 782, and the mildest acute leptospirosis form is in patient infected with 1530 strain. A virulent study were carried out using guinea pig, where they were inoculated with 10 to the power of 8 CFU per mil of the 6 different isolate of leptointerrogan strains, and they were observed for 14 days. And the liver, lung, and kidney were taken for histopathology examination. And the findings of histopathology has confirmed that the result were concordant with the previous severity score, which was Langkawi had caused the most severe acute leptospirosis with total area of hemorrhage in the lung is around 25%. And the next most virulent strain is 782, followed by 898, 1489, and 1548 respectively. And strain 1530 has caused the mildest acute leptospirosis. For the second objective, serotyping was done to classify the isolate into serogroup and serovars, and it was noted that 3 out of 6 isolate were classified as icterohemorrhagic serogroup, 2 Batavia and 1 Canicola. And for the MLST, strain 898 and 1548 agreed well with the findings of serotyping, whereby the other strains were assigned with a novel ST, and there were also novel allele being noted for Langkawi and 1530. So this study, the isolates of this study are representative of the 2 most common serogroup that is endemic in Malaysia, i.e. icterohemorrhagiae and Batavia. So next, a WGS typing were constructed to see the inter-strain relatedness using high-resolution typing, and 8 gene bank isolates with 15 archived Malaysian strains and 6 clinical isolates were assigned for WGS, whole genome sequence, and two types of WGS typing were constructed, i.e. core genome MLST using redome seq-sphere and SNP-based genotyping using parsnip harvest. And it is showed that SNP-based genotyping have higher reference genome coverage. And looking at WGS typing versus MLST, both WGS typing, i.e. CG-MLST and SNP-based genotyping agreed well to ST clustering and also highly congruent to MLST. Between CG-MLST and SNP-based genotyping, both had higher resolution. However, a SNP-based genotyping has better discriminatory power. And among the 6 isolates, 782 and Langkawi are the closest, which suggests a closely related gene content. This had come up to a hypothesis that WGS typing may predict virulent potential. Okay, next, for the third objective, is the genome characterization, whereby all genomes were closed using PacBio and Illumina sequencing except for strain 1530. This is due to higher DNA fragmentation in the plasmid of 1530 that couldn't be resolved. And all genomes contain 2 chromosomes, i.e. chromosome 1 had a substantial genome information around 4.28 to 4.56 megabase. And chromosome 2 only comprised of 350 to 395 kb. The largest genome in this study is the 1489, which is enlarged by the presence of 7 plasmid. And the size is around 5.3 megabase. 898 is the smallest genome without any plasmid. Next, we studied the core genome by analyzing it using protein ortho, and the core genome consists of 3,318 CDS, which compose of 77 to 90% of total CDS in the 6th genome. And the strain-specific CDS range around 49 to 179. Next, we studied the large collinear blocks, which is to see the homologous region of sequence shared by the genomes of the 6 isolates. And this was carried out using mouth aligner software. And the LCB correspond to conserved synteny regions that is represented by the colored boxes. And in this study, we found that there were major genomic rearrangement found in chromosome 1, which represented by line connecting between the strains. And insertion sequence were also found in the junction of rearrangement, which suggests homologous recombination has taken place. And all genomes are widely collinear and synteny. Next, we studied the RFB locus, which encodes the O antigen of Leptospira interrogans, and this is associated with serovar identification and pathogenicity. RFB locus of Icterohemorrhagia serogroup strain is more conserved compared to the rest. And RFB locus of Batavia serogroup strain is shorter with 4 to 10 genes and contain consecutive transposases, which suggestive of genome plasticity. 782 had the longest RFB locus with 2 major gene clusters. And interestingly, Langkawi has 2 extra genes that is differ from the strains in the serogroup. Next, we studied plasmid distribution using plasmid finder, and plasmid are commonly found in Leptospira. Only up to 20 kb of the plasmid were identified in the gene bank, and more than 10 novel plasmid were identified in the study. They are associated with replication, recombination, and also repair. Next, insertion sequence were studied using IS Finder, and more than 20 types of insertion sequence were found in all 6 genomes. A majority is 16 is IS-Lin1, which is highly found in the Batavia serogroup. And it is hypothesized that IS would facilitate host range switching evolution and diversification of Leptospira species genome. As a conclusion, this is the first study that correlate differential clinical severity of Leptospira interrogans with genome analysis. And this is also the first study that used WGS typing of Leptospira, i.e. CG-MLST and SNP-based genotyping. Novel findings in the study include there were 4 novel ST identified in the MLST and more than 10 novel plasmid were found. Langkawi is the most virulent strain, whereas 1530 is the least virulent. However, the RFB locus and the genomic characterization of these two strains are comparable to other strain in the serogroup Heterohemorrhagiae. And Langkawi and 782 are closely positioned in WGS typing, which suggests similar gene content. And it is hypothesized that WGS typing may predict potential virulence. Next, WGS typing delineate Leptospira interrogan strains at higher resolution, and they are highly congruent to MLST clustering, and the SNP-based genotyping have a higher discriminatory power compared to CG-MLST. Finally, genome plasticity is mostly driven by plasmid and IS element that give rise to homologous recombination and horizontal gene transfer. They are also associated with ability to adapt and survive in different hosts and environment. And genoplasticity is highly found in the Batavia serogroup strains that is highly flexible and no known specific carrier. This is the findings. The rest of the findings of my study are being published in this manuscript. And I would like to acknowledge my co-investigator from 4 different countries, that is Malaysia, Germany, Poland, and Netherlands. The study is being funded by Ministry of Health Malaysia. With that, I thank you.
Thank you, Dr. Siti, for the interesting talk. If there's any question that you would like to ask about this presentation, kindly submit your questions using the link provided in the YouTube channel. Now, our second speaker is Puan Jama'ayah Mohamed Zahidi, who is a research officer in Bacteriology Unit, Infectious Disease Research Centre, IMR. She holds a degree in microbiology from University Putra Malaysia. Recently, she has just completed her Master of Science in Molecular Biology from University Technology Mara. Her research area of interest are zoonotic diseases, in particular Brucella and Rickettsia, genomic study and shotgun proteomics aspect. She has published several papers in international and peer-reviewed journals. It is our great privilege to have Puan Jama'ayah with us today to share her research. Her presentation is entitled Comparative Analysis of Genetic Relatedness of Brucella melitensis in Malaysia. Over to you, Puan Jama'ayah, or the screen is yours.
Thank you, Ms. Chairperson, Dr. Rahida, to introduce me briefly. And very thank you to organizing committee that invite me for these sessions. Assalamualaikum and very good morning, ladies and gentlemen. Today I am presenting a part of my study that use whole genome sequencing analysis with the title Comparative Analysis of Genetic Relatedness of Brucella melitensis in Malaysia. This is the outline of my presentations. Let's start with the brief introduction about the disease. The disease is known as Brucella disease or brucellosis. So it is a zoonotic disease which caused by the bacteria of Brucella species, which usually transmitted to the contacted infected animals. Contacted infected farms animals or by the consumed and pasteurized dairy products. There are 6 classical Brucella species, which are Brucella melitensis, Brucella abortus, Brucella suis, Brucella canis, Brucella ovis, Brucella newtoni. Recent years, another 6 species was discovered, which the most hosts come from the frog, fish, and rodent. Of the 6 classical Brucella species above, 4 species are very pathogen. However, Brucella melitensis is very highly pathogenic strain as compared than others and carry very low low infectious dose organism with highly laboratory infectious exposure. This is a global distribution of human brucellosis. The WHO has been reported about 500,000 new cases of human brucellosis in Asia, and the disease can be found in the Mediterranean, Middle East Africa, and Latin America. So, because of the increasing of the number of Brucella melitensis, uh, sorry, so this slide is show for Brucella cases in Malaysia in IMR deposit from the 2009 to until 2021. So as an increasing numbers, human brucellosis cases in Malaysia in 2011 was recorded. For examples, in that year, there is an outbreak occurred in the Penang, which reported by Leong et al., which affected in about 79 people infected to Brucella melitensis due to the consumed unpasteurized goat's milk. Then 4 years later, there is outbreak also occurred in 2015. Presence in Brucella melitensis in Johor, which 17 isolate samples were sending for to IMR for confirmations. Brucella melitensis is a gram-negative bacterial. It is a cocobacilli or short rods, and there is a non-motile and very fastidious intracellular organism. Natural hosts of these bacterial melitensis are sheep and goats. The Brucella melitensis consists of 3 biova, and most human infections caused by the biova 1, while all the 3 biova can be seen in East Mediterranean lineage. Generally, a genome size of Brucella species approximately we can found in 3.3 megabits and consists of the 2 chromosome, which contains about 57%, and there is no plasmid found. A whole genome of reference genome Brucella melitensis 16M was first described in 2002, consists of 2 circular chromosome 2 of 2.11 MB, chromosomes 1 and 1.18 MBP in chromosome 2. Traditionally, of identification and typing of Brucella melitensis is done by biotyping techniques and required hazardous handling of live bacterial. The MLST and MLVA are commonly used in epidemiological investigation. This test is a gold standard for molecular epidemiology for molecular typing, whereby this technique is not sufficiently sensitive to detect new mutation due to the Brucella is very highly conserved. Therefore, WGS, whole genome sequencing analysis, will be able to give better resolution for genotyping and provide a powerful tool for intraspecies discrimination among of Brucella, including relationship to geographical source, any prediction of antimicrobial resistance and protein prediction. Because the increasing number of Brucella melitensis cases passed in 10 years, we can see the lacking of genomic data of Brucella isolates between human and animals made it difficult to conduct traceback epidemiological investigations. Without this data, disease management, both human and livestock, is limited and difficult to conduct a follow-up traceback study if any outbreak or sporadic cases. Therefore, robust genotypic data needed to distinguish the different genotypic among Brucella melitensis isolates from sporadic and outbreak. There is a objective for this study is firstly to determine the genetic relatedness of Brucella militensis isolates from sporadic human infections in Malaysia compared to strains from other locations. Second is to genotype the Malaysian sporadic Brucella militensis strains using MLST. As a sampling, 25 isolates of Brucella militensis were collected from Institute for Medical Research between 2011 to 2016 of sporadic cases from various parts of Malaysia, including Perlis, Penang, Perak, Pahang, Selangor, Kuala Lumpur, Putrajaya, and also Melaka and Johor. For comparative genomic analysis, a total of 75 complete and incomplete genome assemblies of Brucella melitensis was retrieved from the NCBI database. To analyze we include a reference genome Brucella melitensis 16M. To achieve both objectives, the 25 strains of Brucella melitensis will proceed to the genomic sequencing. So all the 25 isolates will carry out about 48 hours culture, which handle in the biosafety cabinets, appropriate PPE in BSL3 and perform DNA extraction. Then we proceed to library preparations and sequence the genomic using Illumina MiSeq platform. Then the generated raw reads of sequence will be filtering and cleaning using FastQC platform. Then assemble and annotate the sequence using the SPADES and RAS toolkit. Then from the assemble sequence, will be analyzed the phylogenomic analysis using the k-SNPs, and as well as we detect in silico MLST using the PUP MLST. Yes. So we come to the results. As a result, in genomic sequencing, firstly, this figure revealed that for quality of sequence reads, which assess using FastQC. So all the quality sequence will trim and to improve overall quality of the sequence and depth coverage. Then the sequencing reads were assembled de novo using SPADES. As a finding, most of the strain generate between 20 to 31 contigs with average genome size about 3.2 mbp. While the GC%, these genome are consistent with the found 57.24 and none of the strains contain plasmid. This result also consistent with other published Brucella melitensis genome and reference Brucella melitensis 16M. While annotated genome of 25 strain Brucella melitensis contains protein coding sequence about between 3,296 to 3,371, while 52 transfer RNA and 3 ribosomal RNA. This pie chart showing a total of gene in 25 strains of Brucella melitensis consists of 42,239 genes in different subsystems where average of each strains between 1,719 and 1,756. Finding of whole genomes phylogenetic analysis was used to determine genetic relatedness between 25 Brucella melitensis strain in this study, with other strains collected from the diverse geographical location. This phylogenetic tree obtained from the SNP-based in relation to 5 geographic cluster, namely genotype 2 West Mediterranean, while genotype 1 is West Mediterranean, while genotype 2 is Mediterranean, genotype 3 is African clade, in European clade, genotype 4, and genotype 5 is American clade. So genome phylogeny show that all 35 Malaysian strain, which included 25 sporadic isolates in this study and 6 humans isolates from other studies and 3 animal isolates grouped together within East Mediterranean in genotype 2. In our analysis showing the genotype 2 comprise about 74 strains of 99 Brucella melitensis and represent the largest Brucella melitensis genotype. Most of the genotype two are from diverse geographical location including Malaysia, Thailand, South Asia, Indian, Pakistan, East Asia, China, Western Asian, Saudi Arabia, Iraq, and Europe. Of the 33 out of the 34 Malaysian strains form a single clade class support by 100% bootstrap value. Interestingly, we found that BMM36/11 is our local strains. Is slightly different lineage clade from Malaysia, but is still clustered in the genotype 2. From the SNPs analysis also, we findings show that one strain from the Philippines, which is Phil 1136 12/1, is shown cluster within the Malaysian clade, indicating a high degree of similarity with our Malaysian strains. While this topology showed only genotype 2, East Mediterranean, so we can see that strain BMM36/11 is unexpectedly show a different lineage clade from the Malaysian, as the strain was clustered more closely to the strain Brucella melitensis biova 3 int from the India, and Brucella melitensis BWIM strains from the Saudi Arabic, and Brucella melitensis 1C is from the Norway. So this SNP phylogenetics results suggest that there is a highly related between the local strain, our local strain BMM36/11 and strain from the Saudi Arabia. In addition, this strain was isolated from the patients that presents a recent infection, a recent history of travelling to the Middle East, and had consumed and pasteurized goat's milk while the travelling. So this is a SNP. The table is shown that SNP matrix number between 34 Malaysian strains. So based on the previous phylogenetic tree slides that we shared, it can be observed that the gene sequencing among the Malaysian Brucella melitensis strains are highly conserved. And these 34 Malaysian strain of Brucella melitensis having the SNPs number of SNPs range from 2,228 to 2,242 when compared to reference genome Brucella melitensis 16M. Besides that, we know that BMM36/11 is for our local strain showed the clustered outside of the main group and is positions to the outlier branch together with the reference strain 16M. Okay, for the in silico detection MLST, we analyze using PubMLST, which use 9 and 21 loci housekeeping gene. So from our analyzing using the whole genome MLST, 24 of the sporadic Brucella melitensis was assigned to type ST39 using the 9 loci. while BMM 36/11 was assigned to ST8. And we found that 4 strains from the study that found a new MLST, new number, which we sent to the curator, and it is a novel and is a ST160. So therefore, our finding using whole genome MLST able to trace the origin of Brucella melitensis. So as a conclusion, phylogenomic analysis using SNPs and whole genome MLST showed that Brucella melitensis strains in Malaysia can be differentiated from strains of other location and clustered into closely specific clade within the East Mediterranean genotype 2. It is an important conclusion that applying the genomic sequencing analysis provide useful data to help identify source of origin of particular strain, which beneficial to the public health researcher and scientists. Last but not least, I would like to express my deepest gratitude to my co-investigator and my collaborator from the UITM, University Technology Mara. With that, I end my presentation. Thank you.
Thank you Puan Jama'ayah for the insightful presentation. Okay, now our third speaker is Puan Hana Farizah Zamri, who is a research officer in Bacteriology Unit, Infectious Disease Research Center, IMR. She holds a degree in Biomedical Science and Master of Science, Medical Microbiology. Both are awarded by University Putra Malaysia. Her research area of interest are tuberculosis genetics, pharmacokinetics and pharmacodynamics of antibiotic. She has published several papers in international and peer-reviewed journals. It is our great privilege to have Puan Hana with us to share her research. Her presentation today is entitled Usefulness of Whole Genome Sequencing in Genotyping Mycobacterium Tuberculosis in Malaysia. Over to you, Puan Hana. The screen is yours.
Thank you, Madam Chairperson, for the introduction. Thank you to the organizing committee of the IMR-SEAMEO webinar for having me here. For my presentation today entitled Usefulness of Whole Genome Sequencing for Genotyping Mycobacterium Tuberculosis in Malaysia, I'll be talking a little bit on Mycobacterium tuberculosis, drug resistance and genetic determinants of resistance in Mycobacterium tuberculosis, and finally go into application of whole genome sequencing in tuberculosis research in Malaysia. Mycobacterium tuberculosis, also known as MTB, is an acid-fast bacterial species within the Mycobacterium tuberculosis complex. It is a causative agent of tuberculosis, better known as TB. It usually attacks the lungs, causing pulmonary tuberculosis, but it can also affect other body sites, causing extrapulmonary tuberculosis. It does not stain very well with the gram stains and appears as a weakly gram-positive bacilli. It stains very well with the acid-fast stains, showing bright red bacilli under the microscope. It has a genome size of around 4.4 million base pairs, encoding for approximately 4,000 genes. The MTB strains are divided into several different lineages according to their geographical distribution. These are the 6 major lineages. Robust classification of these lineages is important not only for taxonomy purposes, but it has been shown that different lineages may carry difference in virulence and antibiotic resistance. Okay, drug-resistant tuberculosis, generally there are several types of resistance. Rifampicin resistant is when the TB is resistant to the first-line antibiotic against tuberculosis. It's called rifampicin resistant TB. Multidrug resistant TB is when the organism is resistant to both rifampicin and isoniazid, 2 of the main anti-tuberculosis agents. And there is the extensively drug-resistant tuberculosis or SDR-TB. This is when the MDR-TB resistant to both rifampicin and isoniazid also resistant to the fluoroquinolone antibiotics and also second-line injectable drugs. So drug-resistant tuberculosis develops most commonly through genetic mutations. Rapid molecular tests are recommended by the WHO and they endorse the Expert MTB, RIF Ultra and TrueNet assays. These assays are automated assays using PCR to detect specific resistance mutation towards rifampicin. So what are the genetic determinants of drug-resistant tuberculosis? As I mentioned earlier, DRTB or drug-resistant tuberculosis is caused by mutations in the genes associated with antibiotic targets. For example, rifampicin resistance occurs when there is mutation in the RPOB gene. Isoniazid resistance occurs when there is mutation inside CATG or in INHA gene, and so on and so forth. In multi-drug resistance, you can find multiple mutations in multiple genes. This is a list of all the drugs used to treat TB and the genes involved in resistance. Okay, if you Google TB whole genome sequencing, you will get hundreds of thousands hits because this is quite a popular field of study. But not only for research, it is now increasingly being used by global TB programs to monitor transmission, to identify potential outbreaks, and for the surveillance of drug resistance. Major health organizations such as the CDC uses whole genome sequencing and recommends the use of whole genome sequencing in their fight against tuberculosis. Also, the UK Health Security Agency under the UK government uses whole genome sequencing for investigating the cluster and transmission occurrence. And the WHO has come up with a catalogue of the thousands of mutations that occur in Mycobacterium tuberculosis and their association with drug resistance. Okay, what about TB whole genome sequencing in Malaysia? You cannot find a lot of, relatively not a lot of publication, but we have several good publications, mostly from the universities, and they characterize drug-resistant and drug-susceptible TB. They use whole genome sequencing to study genomic epidemiology of tuberculosis in Malaysia and to compare the diverse lineages and also drug-resistant populations. We also have characterization of the rare, extensively drug-resistant Mycobacterium tuberculosis in Malaysia. What about IMR? So prior to the COVID-19 pandemic, IMR and the Institute for Perubatan Respiratory, IPR-KL, collaborated in a TB whole genome sequencing study. The objective of the study was to investigate lineage and resistance genotypes circulating within a drug-resistant MTB population in Kuala Lumpur and Selangor. We collected 56 clinical MTB isolates with known resistance towards rifampicin between the years 2017 and 2019. Whole genome sequencing was used. We used the MySeq sequencer from Illumina, and sequence analysis was done using online whole genome sequencing genotyping platforms specific for the TB genome, namely Farizah, TB Profiler, and Microbe. These are available freely online. Very good and very user-friendly, especially for the non-bioinformaticians. Okay, our first findings. When we compared the lineage distribution, we found that this East Asian Beijing predominated with 71.4% of the population, followed by the Indo-Oceanic lineage, Euro-American lineage, and a little bit East African Indian caste lineage. Out of the major 71.4% East Asian Beijing strains, which are resistant to rifampicin, 65% of them were found to be multi-drug resistant, meaning resistant also to isoniazid. Studies by Ismail et al. In 2014 and Farizah et al. In 2020 showed the predominance of Indo-Oceanic strains in a mixed MTB population. But when we look closer to their data, we found that within the drug resistance isolates, there was a higher prevalence also of the East Asian Beijing isolates. This is in accordance with many studies showing that Beijing clones are more prone to carry multiple resistance and are the most widespread resistance clone in Southeast Asia. One of our findings was detection of the extensively drug-resistant TB. This isolate showed phenotypic drug resistance that were resistant towards isoniazid, rifampicin, and streptomycin, and susceptibility towards ethambutol. No results for the remaining anti-tuberculosis agents were obtained due to repeated contamination. whole genome sequencing detected resistance-causing mutations to all of the first-line drugs, including the ethambutol, to all the fluoroquinolones, and to second-line injectable agents, capromycin, amikacin, and kanamycin. This would be the fourth XDR-TB reported in Malaysia, the third to be characterized and published, the first two being previously reported in 2015 publications by Kwan et al. And Ali et al. Another of our findings, when we focus on the rifampicin resistance, for rifampicin resistance, this occurs when there is mutation within the rifampicin resistant determining region, which is the RRDR. This sits between codon 426 and codon 452 within the RPOB gene. So we found multiple resistance in multiple sites, and in one site we have multiple combination of resistance. It is not common to find resistant mutations outside of the RRDR region, but in this study we found 4 such isolates. This is a list of all the mutations found and the frequencies in our MTB population sample. Okay, investigating potential TB transmission. For example, in our study, we found 2 rare Latin American Mediterranean sublineage, only 2 of them in all the 56 isolates. Both of these come from different patients. They share the same susceptibility pattern and also share a rare isoniazid resistant gene mutation that is not present in any of our other isoniazid resistant strains. They also have the same treatment history. They started treatment in 2014, finished their medication, and had reactivation of TB in 2017. One was an unemployed non-Malaysian housewife, while the other was a local Malaysian resident, resided within a 12-kilometer radius of each other. So this is an example of how whole genome sequencing can point towards transmission investigation. All of the results presented here has been published in the Journal of Global Antimicrobial Resistance. It is available online. So to conclude, there are various emerging mutations in the community with undetermined resistance association and limited available data in Malaysia. So whole genome sequencing is useful in inferring and predicting drug resistance, in determining diverse lineage and sublineages of our circulating MTB, and in investigating drug resistance progression and transmissibility of MTB. I would like to acknowledge my team members from IMR, Farizah, Ahmad, and Siti, Roszilawati, Ramli, and also from IPR, Cik Izai Nur Harusan. With that, I end my presentation. Thank you for your attention. Back to you, Madam Moderator.
Thank you, Puan Hana, for the insightful presentation. We will now be moving to question and answer session. However, in the interest of time, I can only entertain 1 questions from the audience. Nevertheless, you can still post all your questions in the chat box, and our speaker will try their best to answer them through emails. Okay. This question goes to Dr. Siti. From the information and analysis provided in your PowerPoint, patient infected by strains labeled Langkawi and 782 were associated with a more severe form of leptospirosis. In the animal study as well, this strain seems to result in earlier signs and symptoms and produce significant histopathological changes upon examination of the tissue. Is that peculiar virulent gene present in this strain that is responsible for the more aggressive clinical disease? I invite Dr. Siti to answer the questions.
Thank you, Madam Moderator. For this study, unfortunately, there is no significant genes that is significantly associated with the severe phenotypic features in Langkawi and 782. We did analyze the 13 confirmed virulence factors in Leptospira interrogans. However, all genes were conserved in all strains, and we did also blast the genomes in the virulence finding database, and there were no significant genes that were presented in the Langkawi and 782 that differ from other strains. But interestingly, there were sets of genes in Langkawi that we found to be upregulated. But however, this study, this is found in a transcriptomic study, and it won't be discussed here.
Okay, thank you, Dr. Siti, for the answer. Maybe I can allow just one question for Puan Hana. The question goes, is there any targeted NGS being conducted for TB research in Malaysia? Puan Hana, I invite you to answer the questions.
Thank you for the question. Yes, there are actually several, I think, tests for target NGS, and one of them is being conducted in IMR, headed by our first speaker, Dr. Siti Roszilawati Ramli. In fact, we are trying to, this will help in specific detection of drug resistance, and with the hopes of cutting down the costs and maybe trends, and maybe we can put it in to use in diagnostic settings. So yeah, you can contact Dr. Siti Roszilawati if you are interested to know more.
Okay, thank you, Puan Hana, for the answer. The question actually came from Puan Aimi Zafira. Thank you, Puan Aimi. She is from University Tan Jimara. I'm afraid that is all the time we have for the questions from participants. Any additional questions, please will be answered by our speaker through email. Kindly leave your email in the chat box when you post your questions. We hope that this webinar has shed lights on some research work that we are conducting at our institute, and would spark interest and initiate research collaboration with others. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session successful. You may subscribe to our National Institute of Health YouTube channel and click for notifications whenever there is a new post. Before I conclude, I would like to announce that we will have the next session of IMR-SEAMEO webinar series on the 16th December 2022. So do mark your calendars. The theme for the next webinar is Akarai, Tiny Creatures with a Significant Health Risk. We hope you can join us at the next session. Until then, many thanks from us at IMR. Take care and stay safe. Thank you.